Have you ever looked at your family and wondered, why do I have my grandmother's nose, but maybe my father's laugh?
Mm-hmm.
Or perhaps, why am I so different from my siblings, even though we share the same parents?
Well, today we're diving into the incredibly complex blueprint that makes you, uniquely you, human heredity. Our mission in this deep dive is to really unpack the core anatomical structures and physiological functions related to how traits get passed down. We're basing this on the fascinating Chapter 29 of Human Anatomy and Physiology, the 10th edition. We'll be pulling out the most important nuggets, looking at figures, clinical applications, body systems, all that good stuff. Get ready for some genuine aha moments.
Absolutely, and it's a journey that, scientifically speaking, really kicked off way back in the mid-1800s with Gregor Mendel.
Yeah.
He was in his monastery garden meticulously studying these either-or traits in pea plants. Simple beginnings, really.
Right, the people. Yeah. But our understanding has just exploded since then, especially with huge efforts like the Human Genome Project. They mapped the entire human DNA sequence, which was monumental. And this research holds immense promise, for much better genetic screening, for developing targeted drugs. It's really pushing medicine forward.
Okay, so to navigate this deep dive into what makes us who we are, we first need the right language.
Yeah.
The basics of genetics. Let's start right there. What are genes, fundamentally? How do they act as these sort of recipes for us?
That's the perfect starting point. Genes are specific segments of DNA. Think of them like detailed instructions or genetic blueprints, instructions for building proteins. Well, they do almost everything in your body. They dictate your molecules, your characteristics, hair color, blood type, even your genetic sex.
Okay, the instructions for everything. And where are these instructions kept?
They're located on chromosomes. Now, in almost all human cells, sperm and egg cells are the exception, we have 46 chromosomes. That's the diploid number. They're organized into 23 pairs of homologous chromosomes. You get one chromosome in each pair from your mother and one from your father.
Homologous meaning similar.
Similar, yeah. They carry genes for the same traits. Now, it's key to distinguish the two sex chromosomes X and Y. They determine sex from the other 44 chromosomes. Those other 44 are called autosomes, and they carry the vast majority of your other genetic information.
So we have these chromosomes packed with genes. How do scientists actually see them? Check if everything's there. What's a karyotype?
The karyotype. Figure 29.1 shows one nicely. It's basically a picture of a person's full set of chromosomes all lined up in their homologous pairs, like a genetic inventory.
How do they make that picture?
Well, scientists take cells, usually white blood cells called lymphocytes, grow them, and then stop them mid-division when the chromosomes are condensed and visible. They stain them, photograph them, and then often use computer analysis to arrange them by size and shape into those pairs.
And that tells you.
It's a really powerful diagnostic tool. You can immediately spot if there's an extra chromosome, like in Down syndrome, or a missing one, or even bits that are swapped or deleted. Big clues to genetic disorders.
Okay. So zooming in on those chromosomes, on a specific gene, you mentioned variations. That gets us to alleles, right? What are they?
Exactly. Alleles are just the different versions of a particular gene, like different flavors of the same instruction. They're matched genes found at the same spot, the locus on homologous chromosomes. They code for the same trait, but maybe different forms of it.
Like the thumb ligament example.
Perfect example. One allele might code for tight thumb ligaments, another for loose ones, the so-called double-jointed thumb.
And depending on which alleles you get, we use terms like homozygous and heterozygous.
No.
Can you break those down?
Sure. If the two alleles you have for a trait are identical, say you got the tight ligament version from both parents, we might write that as JJ, you're homozygous for that gene. But if the alleles are different, maybe you got J for loose ligaments from one parent and J for tight from the other, then you're heterozygous. That's JJ.
Okay, that makes sense. Which leads right into dominant and recessive alleles.
Yeah.
How do we know which allele wins, so to speak?
Right. A dominant allele, we use a capital letter for it. Like J is the one that masks its partner. It shows up in the phenotype, whether you have one copy JJ or two JJ. So if you're JJ for thumb ligaments, you'll have the double-jointed trait because J is dominant.
And recessive.
A recessive allele, lowercase letter, like J only gets expressed if you have two copies of it. So only the J genotype results in tight thumb ligaments. The dominant J overrides it otherwise. So putting it all together, genotype is your actual genetic makeup, the specific alleles you carry, like JJ. And phenotype is how that genotype is physically expressed, the observable trait, like actually having double-jointed thumbs.
Wow.
And what's really key here, these terms, gene, allele, homozygous, heterozygous, dominant, recessive, genotype, phenotype, They're not just words. They're the fundamental building blocks for understanding everything else about inheritance.
Okay, so we've got the basic language down, but here's where it gets really interesting. If we all have pretty similar genes, why is each of us so unique? I mean, apart from identical twins, maybe.
That's the beauty of genetic variation. That's why you're not a clone of your sibling. This amazing uniqueness comes down to three main events happening before birth.
Three key things.
Yep. First, the independent assortment of chromosomes when sperm and eggs are made. Second, crossover between homologous chromosomes, which shuffles genes around. And third, good old random fertilization.
Okay, let's unpack that first one. Independent assortment. What exactly is happening there?
So during meiosis, that's the cell division that produces gametes, the replicated homologous chromosomes pair up. They form these structures called tetrads. Now, how these tetrads line up on the center line, the metaphase spindle, before they get pulled apart, it's totally random, pure chance.
So which parental chromosome goes into which sperm or egg is random?
Completely. It leads to a random distribution of your maternal and paternal chromosomes into each gamete. Each sperm or egg gets a unique mix. And just as a side note, errors in this precise shuffling are sometimes linked to conditions like Down syndrome, where there's an extra chromosome 21.
And the sheer number of possibilities from just this one step is huge.
Oh, it's astronomical. For a simple organism with just, say, 6 chromosomes, you get 8 different gamete types. But for us humans with 23 pairs, independent assortment alone creates 2 to the power of 23 possible combinations. That's about 8.5 million different types of sperm or egg cells one person can produce.
8.5 million. Wow. Okay. And then there's a second factor, crossover and gene recombination. Figure 29.3 helps visualize this. How does that add even more variety?
Right. So genes on the same chromosome are usually inherited together, they're linked, but it's not absolute. During meiosis, when those homologous chromosomes are paired up in tetrads, they can actually swap segments. They break and exchange pieces.
They trade parts.
Exactly. Imagine your dad gave you a chromosome with alleles for blonde hair and blue eyes, and your mom gave you one with brown hair and brown eyes. During crossover, a piece might swap, so you end up with a chromosome carrying blonde hair and brown eyes or brown hair and blue eyes. These are new recombinant chromosomes.
So it mixes up the combinations on the chromosome itself.
Precisely. It creates totally unique combinations of alleles that weren't on the original parental chromosomes. This happens across all 23 pairs of chromosomes. The variability introduced by crossover on top of independent assortment is just, well, it's immense. Almost impossible to calculate fully.
Mind-boggling, really.
And then the final layer is random fertilization. You've got gametes being produced with all of this incredible variety. And then which one specific sperm out of millions fertilizes which one specific egg? It's completely random, a total lottery.
So combine independent assortments, 8.5 million options from the egg and 8.5 million from the sperm.
You're looking at one offspring representing 1 out of roughly 72 trillion possible zygote combinations, even before you factor in the extra variation from crossover.
72 trillion.
Yeah, and that's the big picture takeaway. This incredible genetic lottery is why siblings, sharing the same parents, can look and be so different, yet still obviously share family traits.
Okay, amazing. So we have the vocabulary, we have the sources of variation. Now, how do these principles play out in passing down specific traits? It's not always straightforward, is it? Let's start with dominant recessive inheritance. How do we figure out the odds?
Right. For these simpler cases, we use a tool called a Punnett square. Figure 29.4 shows one. It helps predict the possible gene combinations in offspring if you know the parents' genotypes. Let's use tongue rolling. Ability to roll is dominant. T, non-rolling is recessive.
G.
If both parents are heterozygous, TT, the Punnett square shows the possibilities.
Okay, what does it show?
It shows a 25% chance of getting TT, homozygous dominant roller, a 50% chance of TT, heterozygous, also a roller because T is dominant, and a 25% chance of TT, homozygous recessive, non-roller.
So phenotype-wise, that's a 75% chance of being in a tongue roller and 25% chance of not being one.
Exactly. But remember, these are probabilities, like flipping a coin.
Right, not guarantees for any single child.
Precisely. The more offspring you look at, the closer the actual ratios tend to get to the predicted ones. Each conception is a totally independent event. Having one teat child doesn't change the odds for the next one.
And Table 29.1 lists some common dominant traits. Widow's peak, dimples, freckles, things we see all the time.
Yeah, those are good examples. Now, interestingly, disorders caused by dominant genes are actually quite rare.
Why is that?
Usually because if a dominant gene causes a lethal condition, it's expressed early, and the individual often doesn't survive to reproduce. but there are exceptions. Huntington's disease is a key example. It's a fatal nervous system disorder caused by a dominant gene, but it's a delayed action gene, meaning symptoms typically don't appear until around age 40. So individuals can have children before they even know they have the gene. Their offspring then have a 50% chance of inheriting it.
That's tough. Okay, so that's dominance. What about traits that only show up if you get two copies? Let's talk about recessive traits.
Right. Many, maybe even most genetic disorders are actually inherited as simple recessive traits. Things like albinism, cystic fibrosis, Tay-Sachs disease. For these, individuals called carriers are heterozygous. They have one copy of the normal allele and one copy of the recessive allele for the disorder.
So they don't have the disease themselves.
Correct. They're usually phenotypically normal, healthy, but they carry that recessive allele and can pass it on to their children. If two carriers have a child, there's a 25% chance that child will inherit two recessive alleles and have the disorder.
Okay. Then there's that middle ground, right, where neither allele fully dominates.
Yes, that's incomplete dominance. In this case, the heterozygote has a phenotype that's sort of intermediate between the two homozygous types. The classic human example is the sickling gene for hemoglobin.
Sickle cell.
Exactly. If you're homozygous recessive, you have sickle cell anemia. Your red blood cells distort, causing pain, damage. It's serious. But if you're heterozygous ISS, you have sickle cell trait. You produce both normal and sickling hemoglobin. You're generally healthy, but might have issues in low oxygen situations. And importantly, you can pass the S allele on.
I see. And sometimes there are more than just two versions of a gene floating around in the population.
Absolutely. That's multiple allele inheritance. Even though any one person only inherits two alleles, more than two can exist in the population as a whole. The prime example is ABO blood types.
Right. A, B, ABO.
Exactly. There are three main alleles, IA, IB, and I. IA and IB are interesting because they are codominant.
Codominant.
Meaning, if you inherit both IA and IB, both are fully expressed, resulting in type AB blood. The I allele for type O blood is recessive to both IA and IB. Table 29.2 shows the different combinations and their frequencies.
Okay. Now, what about traits linked to our sex chromosomes? Sex-linked inheritance.
Right. These are traits determined by genes located on the X or Y chromosomes. The key thing here is that the Y chromosome is much smaller than the X.
And it carries fewer genes.
Far fewer. It lacks copies of many genes found on the X chromosome.
What does that mean for inheritance, especially for males?
It means males, XY, are more likely to express X-linked recessive traits. If a male inherits a recessive allele on his X chromosome, like for hemophilia or red-green color blindness, there's no corresponding dominant allele on his tiny Y chromosome to mask it.
So he expresses the trait even with just one copy.
Exactly. Females, XX on the other hand, need to inherit the recessive allele on both of their X chromosomes to express the trait. This is why these conditions are much more common in males and are often passed from mother, who might be a carrier, to son.
Got it. Okay, finally. Most traits aren't just one gene, right? They're more complex.
Absolutely. Most human characteristics aren't simple on-off switches. They fall under polygene inheritance. This means the phenotype depends on the combined effects of several gene pairs, often at different locations on the chromosomes.
And this leads to?
It leads to continuous quantitative variation. Traits that exist on a spectrum like height, skin color, metabolic rate, maybe even intelligence. You see a range of possibilities, often forming a bell-shaped curve in a population, like in figure 29.6.
Can you give an example, like skin color?
Sure. Skin color is thought to be controlled by at least three genes, let's call them A, B, and C, each contributing to dark pigment. Their effects are additive. So someone with AABCC genotype would have very dark skin. Someone with ABC would be very fair.
And heterozygotes.
Heterozygous parents, like ABBCC, can produce offspring with a whole range of intermediate skin tones, depending on the combination of alleles the child inherits. Which really brings up a fascinating point. Looking at all these patterns, how much of who we are is actually set in stone by our genes, and how much can be shaped by, our environment or other genetic controls?
Yeah, that's a huge question. If genotype is the rock, the blueprint, maybe phenotype is more like clay.
Yeah.
Shaped by other forces.
So how can environmental factors actually tweak gene expression, make us different from just our raw genetic code?
It's a really crucial aspect, and it starts super early, even in the womb. Maternal factors can significantly alter development. The classic tragic example is the sedative thalidomide. When taken by pregnant women, it caused babies to be born with severe limb defects, flipper-like appendages.
even though their genes for limb development were normal.
Exactly. These are called phenocopies, environmentally induced phenotypes that mimic genetic conditions.
Wow. So the environment can create a sort of imitation mutation. Does this influence continue after we're born? Oh, definitely. Think about nutrition. Poor nutrition in infancy can seriously stunt growth, brain development, height, overall body size, even if someone has genes that would otherwise predispose them to be tall or have typical development.
So tall genes aren't enough if the environment doesn't support them.
Right. And even other genes can act as part of the environment for a specific gene. Hormonal deficits, for example, like hypothyroidism in childhood can lead to cretinism, which involves abnormal skeletal growth, impacting how height genes are expressed. So the real takeaway here, it's this constant interplay, a dance between nature and nurture. Genetic potential is real, but it's definitely not the whole story of who we become.
But hang on, there's even more complexity, isn't there? More than just the standard genes Mendel talked about, more than just nuclear DNA. What other layers of genetic control are scientists uncovering? I heard something like the protein-coding genes, the ones we usually think about, make up less than 2% of our DNA. What's the rest doing?
That's right, it's a tiny fraction. And it leads us into some really cutting-edge areas. There are at least 2 more major levels of control beyond those protein-coding genes. The second level involves things called small RNAs. These used to be part of what was dismissed as junk DNA.
Junk DNA. Not so junky anymore.
Not at all. We now know these small RNA molecules like microRNAs and small interfering RNAs are incredibly important regulators. They're like mobile control freaks. They can directly interact with DNA with other RNA molecules or proteins to silence genes, stop them from being translated into proteins, or even shut down those pesky jumping genes, retrotransposons.
So they're like gene managers.
In a way, yeah. They're involved in programmed cell death development, and disruptions in them are linked to things like cancers, maybe even schizophrenia. There's huge interest in creating RNA interference drugs to target specific genes. It's a whole new layer of complexity.
Incredible. Okay, tiny RNAs as controllers.
Yeah.
What's the third layer?
The third layer involves epigenetic marks. This isn't about the DNA sequence itself, but about chemical tags attached to the DNA or the proteins it's wrapped around. It's like adding sticky notes or highlights to the instruction manual, changing how it's read without changing the words themselves.
Chemical tags, like what?
Things like methyl groups and acetyl groups, they attach to DNA or to the histone proteins that package DNA. Generally, methylation tends to silence genes, making them unreadable. Acetylation often opens them up, making them active.
And this affects us how?
It's crucial. For example, epigenetics is responsible for silencing one of the 2 X chromosomes in female mammals early in development. It's also implicated in cancer development and potentially many other illnesses. These marks can change throughout life based on environmental factors, too. A really fascinating aspect of epigenetics is genomic imprinting.
Imprinting. What's that?
This is where certain genes are tagged, usually by methylation, in the sperm or the egg, marking them as either paternal or maternal. The embryo then reads these tags and only expresses one copy of the gene, either the mom's or the dad's, while the other is kept silent.
So the parent of origin matters for how the gene works.
Exactly. And usually these imprints are wiped clean and reset in the next generation's gametes, but sometimes they can persist.
Is there a clinical example of this?
Oh, a very striking one. There's a specific region on chromosome 15. If a deletion occurs there, the outcome depends entirely on which parent you inherited that faulty chromosome from. If it's from the father, it causes Prader-Willi syndrome, intellectual disability, obesity. If the exact same deletion comes from the mother, it causes Angelman syndrome, severe intellectual disability, movement issues, happy demeanor. Same genetic deletion, totally different syndromes based on parental origin. Shows the power of imprinting.
That is truly mind-bending. Okay, one more layer. Outside the nucleus.
Right. Extranuclear inheritance, specifically mitochondrial inheritance. We always focus on the DNA in the nucleus, but mitochondria, the cell's powerhouses, have their own small loop of DNA mtDNA, about 37 genes.
And how is that inherited?
Almost exclusively from the mother. The egg contributes virtually all the cytoplasm, including the mitochondria, to the zygote. Sperm mitochondria usually get destroyed after fertilization.
So mitochondrial disorders come from the maternal line?
Typically, yes. These disorders are relatively rare and often involve problems with energy production, oxidative phosphorylation. They can cause muscle or neurological issues, and there's some research linking them potentially to degenerative diseases like Alzheimer's and Parkinson's. So, you see, the bigger picture here is that our understanding of heredity is constantly evolving. It's way beyond simple Mendelian traits. We're learning about all these complex layers of control, which has huge implications for medicine and how we even define heredity.
All this knowledge, it's not just fascinating theory, right? It's a huge real-world impact, especially in health. Predicting and treating genetic disorders. Let's talk about genetic screening and counseling.
Absolutely critical applications. These services help prospective parents understand their risks and options. We already do routine newborn screening for conditions like PKU. Early detection allows for treatment that prevents severe problems.
In finding carriers, people who don't have the disease but carry the gene.
Yes, carrier recognition is key. We can use pedigrees, basically family trees tracking a trait through generations, like figure 29.7 shows for widow's peak to figure out genotypes and probabilities. Plus, we have blood tests, simple ones for things like the sickling gene carrier status, and more complex DNA tests for carriers of Tay-Sachs, cystic fibrosis, and others.
And if there's a known risk during pregnancy, there's fetal testing. What are the main ways that's done?
Traditionally, the two main invasive methods are amniocentesis and chorionic villus sampling, or CVS.
Okay, amnio first, figure 29.8a.
Right. Amnio involves inserting a needle through the abdomen into the amniotic sac, usually after the 14th week, to draw out a small amount of amniotic fluid. That fluid contains fetal cells that have sloughed off. They can be cultured for several weeks, then karyotyped to check chromosomes, or tested for specific genetic markers. Ultrasound guidance makes it safer now.
And CVS, figure 29.8b.
CVS involves suctioning a tiny sample of the chorionic villi, which are part of the placenta, usually done earlier, maybe around 8 weeks. The big advantage is the cells are already rapidly dividing, so karyotyping can be done much faster, almost immediately. But there has been a slightly higher associated risk of limb defects with CVS compared to amnio.
These sound like serious procedures. When are they usually recommended? And is this still the standard?
They're typically offered to women over 35 because the risk of Down syndrome increases with maternal age, or if there's a known family history suggesting a high risk for a specific severe disorder. But honestly, these invasive methods are rapidly being overtaken. By non-invasive prenatal testing, or NIPT, we can now analyze tiny fragments of fetal DNA that circulate freely in the mother's bloodstream. We can get incredibly detailed information about the fetal genome just from a maternal blood draw. It's safer, can be done earlier, and is quickly becoming the new standard.
That's a massive advancement. Okay, finally, what about actually treating genetic diseases? Human gene therapy, where are we with that?
Well, as our diagnostic abilities improve, the focus shifts to intervention. Gene therapy aims to correct or alleviate genetic disorders, especially those caused by a single faulty gene. There are a couple of main strategies being explored. One involves using a modified harmless virus to deliver a working copy of the defective gene into the patient's cells. This has had some success, notably for certain severe combined immunodeficiency disorders, SCID.
Like the bubble boy disease.
Exactly. Another approach is trying to inject corrected DNA directly into target cells. Results have been more mixed here for things like cystic fibrosis or muscular dystrophy. It's still very much a work in progress.
It raises huge questions, doesn't it?
Oh, absolutely. Massive ethical, religious, and societal questions. Who pays for these potentially incredibly expensive therapies? Who decides who gets treated? Are we crossing lines, playing God? As our power to manipulate genes increases, our responsibility to handle that power wisely and equitably becomes paramount. It's a conversation we absolutely need to be having.
We've taken quite the deep dive today into human heredity. From those basic building blocks, genes, alleles, through the incredible ways variation is generated, the different patterns of inheritance, environmental effects, epigenetics, all the way to genetic testing and the frontiers of gene therapy. Considering the sheer precision needed to copy DNA and divide chromosomes perfectly, it really is pretty amazing that genetic mistakes aren't more common. Maybe after this discussion, you have a bit more wonder about how you turned out just the way you did. We really hope this deep dive helps shortcut your way to being well-informed on this fascinating world of human heredity. Warmly thanking you for being part of our last-minute lecture family.